Welcome to Genomics Unlocked, where genomic science and webinars converge. Today's episode covers comprehensive molecular tumor analysis, CMTA, integrating RNAseq and TME for targeted therapy, featuring Dr. Mary Law Yaspo and Priyanka Kurapati.
So good day, everyone. My name is Brett Verdeman, and it is a pleasure to be hosting you all today and welcoming you to our fifth webinar in the Genomics Unlocked series, which is a platform for knowledge sharing in all things genomics driven on DNBSEQ. And today we will explore more on MGI's DNBSEQ technology and Alacris's comprehensive molecular tumor analysis integrating RNAseq and TME for targeted oncology therapy. So today we are proud to welcome Priyanka Kurapati, MGI's field application scientist, and Dr. Mary Law Yaspo, who is the co-founder and chief scientific officer of Alacris Theranostics, who will guide us into the benefits of comprehensive tumor profiling in a clinical setting using Alacris's end-to-end DX service platform and bioinformatics technologies powered by MGI's DNBSEQ technology to improve personalized treatment decisions. So today we are using the GoToWebinar platform. So on your right-hand side of the screen, you should see the toolbar there. And if you have any questions, so there is a questions chat. So just access that if you have any questions for any of our panelists, or you can just direct message to the host, which is myself. And we would leave the questions for the end just to help with the streamlining of the presentations. If you have also not already done so, please register for the on-demand on the genomicsunlocked.com website to access previous webinars that we've hosted before, and also to subscribe to future webinars we'll be having. So thank you all for joining the webinar, Alacris Comprehensive Molecular Tumor Analysis, CMTA, Integrating RNAseq and TME for Targeted Oncology Therapy. So for our first speaker, I would like to introduce Priyanka Kurapati, one of MGI's field application scientists who graduated from the University of Houston, focused on biotechnology, and since then has undergone multiple studies and managed various high-throughput sequences, troubleshooting, validating, and protocols, specializing in multiple NGS applications such as WGS, whole exome, reproductive genetics, pharmacogenomics, and was heavily involved in the population genome programs in the UAE on DNBSEQ T7. So without further ado, welcome, Priyanka.
First of all, thank you very much, Brett, for the introduction. I'm super delighted today to be a part of this webinar and having you all on board today. First off, yes, as Brett mentioned, I'm the field application scientist, and I'm based in Frankfurt, Germany, and responsible for Central East European countries. So today, I would like to talk about the technology, which is unique to MGI, patented state-of-the-art DNBSEQ technology. Before we dive in, I would like to talk a little about what the MGI's vision is. So it has always been leading to the life science innovation, and we all have been thriving to develop and promote the advanced life science tools for future healthcare. So to achieve this, we've got 2,800 employees worldwide, including 36% are research and development employees, and about more than 200 employees are in Europe and Africa, including 70% are technical specialists, engineers, and experienced commercial team. And we have a research and development and production facility in Europe, where it's in the city called Riga in Latvia. And we have about 597 valid patented applications and about more than 2,000 users in 90 countries and regions all around the world. And we also have published papers and in top scientific journals and about more than 4,430 papers. And so far, we've generated data on the MGI sequencing platform, which is almost more than 150 petabytes. So I would like to dive into the technology itself further. So today's agenda is going to be, I'm going to talk about the technology and the MGI sequencers, and the AmpX library conversion. So the DNBSEQ technology. This is a unique DNA nanoball technology where, as you all know, any NGS platform requires a library preparation for our sequencing. So the library preparation involves, like any other, fragmentation of your DNA, and then followed by the end repair, A-tailing, PCR, and then you have your double-stranded DNA. So in MGI, we have an additional step, which is circularizing your DNA to perform the DNA nanoballs. So in order to do that, you denature the double-stranded DNA by heating to 95 degrees, and this single-stranded DNA is further circularized to using the oligos, and this single-stranded circularized DNA is used to make the DNA nanoballs by a process called rolling circle replication. I can show you in the next slide furthermore. And eventually, this DNA nanoball is the product that you use to load onto your flow cell for sequencing. So the rolling circle replication, this is the process that takes place to convert your single-stranded circular DNA library into DNA nanoball in a controlled reaction. As you see in this animation, where this single-stranded is, you see the replication is being performed, and then this is how the DNA nanoball looks like. And one single-stranded DNA repeats about 300 to 500 times in a DNB, and you see a tube full of DNBs, and this mixture is going to be loaded onto the flow cell. So why DNB? Why do we make MGI DNA nanoballs? So here is a comparison of the rolling circle replication and PCR. So when you perform MGI DNA nanoballs, we're using a linear amplification, and which involves very low amplification bias. And as you know, 529 enzyme has the highest fidelity of any known enzyme. So one error in like 10 power 6 to 10 power 7 bases. So you pretty much see like no accumulation of amplification errors, and also low index hopping. So as you see in this picture here, although you have like one error in one of the copy, like it's quite negligible when you're talking about like 300 to 500 copies in a DNA nanoball. Whereas on the PCR, there are a few NGS problems that are introduced when you perform a PCR, where you have the false SNPs, false indels, coverage, and GC bias, and index hopping. So as you see here, if you have an error in the first cycle, you would have it in the furthermore cycles as you keep doing the PCR. And we use a flow cell, which is patterned array, and this technology minimizes the signal cross interference and the combination of the spacing between the spots and optimizing the spot size and the protein embedding. This ensures one DNB attaches to one spot on the flow cell, and it definitely has high density and utilization. In the next slide, I would like to show you how the DNB loading looks like in the real time. But before that, there is a quick video where it shows how the DNBs attach onto the spots. So as you can see, here you go. So they go and bind to each spot, one per spot right there. And this is the DNB loading that takes place in real time. So this is how it's captured by the camera. And finally, you have your flow cell with the DNBs, and this is how the image looks like. So we have different types of flow cells. Like, for example, DNBSEQ G400 has a flow cell which looks like this, and the DNBSEQ T7 has a flow cell with just one lane. Moving ahead, so it is quite efficient to do the way the DNB loading is done, because MGI loads DNBs of the same size on the flow cell with patterned array of chemically modified spots. So what I mean is we optimize the DNB size before we even load. So basically, it's just optimized to the size of the spot. And as you know, DNAs or DNBs are negatively charged. So if there's any other DNB that would want to go and bind to the same spot, it would repel. And this way, there is no signal overlap at all. So I would say like it's almost impossible to overload your flow cell. And finally, the sequencing, and we follow the combinatorial probe anchor synthesis, where the incorporation of the bases happen, and eventually there is the capture of the images, and the base calling is done. And once the base calling is done, the bases are cleaved, and then it goes ahead to the next cycle by the process called sequencing by synthesis. And that's how every cycle is done. And by the end of it, you have the FASTQ files generated that you could use it for further analysis. So the advantages of performing DNBSEQ technology is, firstly, you would see increased accuracy. The reason being you're performing a linear amplification. So you're pretty much using the original strand from the single-stranded circular DNA. And there is almost no error accumulation, so zero accumulation at all. And then you would see decreased duplicates because you're performing the rolling circle replication, which generates like 300 to 500 copies, and there is no overload at all on the flow cell. And moving on, it also reduces index hopping, where there is low PCR bias with these parameters right here. So, talking about the MGI sequencers, you should really experience the quality and affordability with MGI's patented state-of-the-art DNBSEQ technology and genetic sequencers. Well, I believe you all would have heard about the DNBSEQ G400 and DNBSEQ T7, which has been widely used all over the world for years now. But now we also have a new sequencer called DNBSEQ G99, which we've launched this year. And what's interesting about the sequencer is it is the fastest sequencer on planet till date, and it's easy to use sequencer, and it's very much suitable for targeted panels and small genome sequencing. So I would like to show a little about the G99 if you've never seen how it looks like before. So this is the sequencer, so we have the reagent cartridge slots right here. So it is a dual cartridge system, and the sequencing reagents are preloaded, and we also have the cleaning reagents already delivered. And this novel cartridge design involves by just simply pressing to load the reagents into the sequencing cartridge. And G99 comes with two configurations. One is with a bioinformatics module or without. So you could choose how you would want the sequencer to be. So what's interesting about having a built-in bioinformatics module is you can initiate the secondary analysis automatically. And this is a user interface monitor, which is fully animated for real-time visualization. And these here are the flow cell compartments, and you could simultaneously perform 2 runs at a time. And obviously the waste container that you would need for the sequencing. Moving on. So Apex library conversion. Well, this is a very interesting technology where even Murray from Alacris, they've used to perform their third-party libraries to be compatible with the DNB sequencers. So how can we use the third-party libraries onto the DNB sequencers? So what we basically do is we convert the Illumina P5, P7 adapter libraries for MGI sequencers. So the input, or what we recommend, is either 10 nanograms, 25, or 50 nanograms. And there is a PCR reaction that is involved that could be 5, 8, or 10 cycles. So during the PCR, you use P5 and P7 primers, where the P5 primer is with the 5' phosphate. So once you have the P5 primer with the 5' phosphate, like any other circularization like that you do on DNBSEQ, you separate the double-stranded and you get your single-stranded DNA with the 5' phosphate group on the P5 primer. So after denaturation, you have the splint oligo, which is universal and complementary to the P5 and P7 ends, and which ligases, with ligase, it circularizes, and then, you know, you have your single-stranded circular DNA. And once you have your single-stranded circular DNA, you proceed like any other MGI DNBSEQ technology where you perform the DNA nanoballs. And you could use those DNA nanoballs onto the sequencer with Illumina-specific primers. So you would be adding the Illumina-specific primers onto the sequencing cartridge, and it's good to go for sequencing. And we have a wide variety of applications with advanced features, and some of the library preparation solutions were ranging from whole genome, whole exome, whole transcriptome, and then we have RNA applications followed by customized panels, including COVID-19. So we have solutions that fully enzymatic library preparation, and it involves PCR-free options, FFPE compatibility, single and dual indexing, unique molecular identifiers. Finally, I would like to let you know that system is quite scalable, and we have all types of classic experiments, which includes whole genome, whole exome, target sequencing, RNA applications, and methylation sequencing. So all you got to do is choose the sequencing parameters according to the required data and the amount of samples. So furthermore, I would like to give you a take-home message. Obviously, it is the DNBSEQ technology, and we use the DNA nanoball. We use the flow cell, which is pattern array, and the CPAS sequencing for the MGI sequencing. So these three parameters enable you with high accuracy, low duplication rate, and low index hopping rate. So that's all from me, and thank you so much for listening. Now I will pass over to Marie from Alacris, and she's going to talk you through the webinar, the rest of the webinar.
So yeah, thank you, Priyanka, for providing us with some insights into DNBSEQ technology. I would now like to introduce Dr. Marie-Laure Jasper, who is the co-founder and chief scientific officer of Alacris Theranostics, which is a biotech company specializing in the development of innovative approaches for precision oncology. Marie-Laure graduated with her PhD at the Pierre and Marie Curie University in Paris and completed her postdoctoral research at the Imperial Cancer Research in London. She also heads the Otto Warburg Laboratory for Gene Regulation and Systems Biology of Cancer at the Max Planck Institute for Molecular Genetics in Berlin. She has coordinated and participated in multiple consortiums focusing on some of the cancer consortiums, and is currently heading the team at Alacris Theranostics, focusing on translating genomic sequencing and bioinformatic technologies for medical applications. So welcome, Dr. Marie-Laure Jasper.
Thank you, Brett, and hello to everybody. So happy to be with you today, and I will go through our technologies and how we use the MGI technologies in our accredited pipeline for cancer molecular diagnostics. So Alacris is a molecular diagnostics company who has developed pipeline now medically accredited for cancer molecular diagnostics. And we offer, in addition, a wide range of cutting-edge NGS services, including proprietary bioinformatics pipelines and NGS service on metagenomics, also working on the research and development modeling of cancer response to therapies. But back to our talk today, I would focus on the CMTA for comprehensive molecular tumor analysis that we developed to personalized diagnostic for cancer patients. Our company is located in Berlin-Adlershof in the Techno Park, and we operate several robotic platforms in our daily work, and the two sequencers, DNBSEQ G400 and DNBSEQ T7. Our mission at the company is, it's a long haul mission that we have developed from research project and cancer consortium projects. And it can be summarized as advancing precision medicine for oncology with comprehensive tumor profiling. So the comprehensive tumor profiling is getting more and more adoption. What it is about compared to the more canonical or more used panel test is that the panels detect, they're great, they're quick, but they detect only a fraction of the potentially targetable alteration. So that automatically reduces opportunities for personalized therapy. So whereas comprehensive multimodal analysis scans the whole global tumor landscape and maximizes the option to navigate patient-matched therapies. So the navigating the oncology diagnostics, the diagnostics is addressing evolving needs in the precision oncology. So the transition from panels to multimodal test, developing end-to-end accredited pipelines which address more of the new needs for choosing the targeted therapy. It's also adaptable to the different clinical setup and deliver interpreted personalized report. So the comprehensive molecular tumor analysis is at the forefront of the NGS analysis landscape. So there are several options, of course, that you all know from a piece of tissue, a tissue section, is to go for very small panels, and sometimes that makes sense, of course, or to go to exome, which is implemented in a number of clinical outfit. What is less common but becomes more and more needed is a comprehensive bulk analysis of the exome integrated to the transcriptome. Again, to identify more targets for individual treatment and generate valuable data resource for reanalysis, for instance. So precision medicine in oncology is about this, selecting the best treatment based on the understanding of the molecular basis of their tumors. But then to get to this point, to understand the molecular basis and pick the best targets, one has to know the tumor molecular program. The two main inputs for this are mutations, also chromosomal changes, and gene expression levels. So gene expression is immediate readout of the complex genetics and epigenetics. So it gets more difficult to go through the whole epigenetics program, but it's responsible for a large part to the programs occurring in the cancer cell. So why reading out the expression program is important for diagnostics. When we look at the actionability in routine, there are of course different categories of biomarkers. As well understood ones and well known is mutations that are directly targetable, or mutation and alteration which are not directly targetable, but there are biomarkers or signatures supporting a therapy, for instance, a PARP inhibitor for HRD positive tumors. On the other hand, there are other categories of biomarkers which rely on gene expression, which are overactivated or amplified gene targets, or tumor addiction to some markers like BCL2, on the overexpression, it's often ERBB2, CDK4, CDK6. And for those, one has to read out the expression profiles. There are, of course, other biomarkers, and I will come back to these points, the CNV, the gene fusion, and the splice variant. But basically, what I wanted to say is that it is so that the cancer treatment now is following a paradigm shift towards mechanistic drugs. So from more tumor-specific targeted therapies, which are still, of course, ongoing for a specific case, but more and more we see the move to the mechanistic drugs, which target more, as the name says, a mechanism rather than a specific entities. And in this, the role of immuno-oncology plays a large role with a burst of the immuno-oncology drugs. And for instance, the T-cell targeted immunomodulators are now, from the past 2 years, in an very deep explosions. So again, to summarize what I said to why did we develop comprehensive profiling is to address all those needs and to be able to have in one go all this information. So this is a bit more schematics, how the integration of RNAseq information enhance the precision of the diagnostics. So from the DNA alone, even if one do VGS or VGS, which is also done in some precision medicine entities, we have, of course, a lot of information, of key information, which is needed: the mutation, the TME, the HRD, the structural alteration, and the copy number variants. But the hidden part of the iceberg is actually a very rich information that we nowadays need to access more and more, which are the predictive signature of drug response, signature of tumor subtypes. There are a number of expression profiles which allow to characterize the tumor in subtype, go to the estimating the tumor microenvironment by, I will come back to this, reading out the RNAseq and deconvolving what makes the tumor microenvironment. And of course, all the other aspects which are equally important: gene fusions, splice variants, and other expression signatures. So the CMTA, as it is, by integrating exome and transcriptome, enables personalized therapy for all tumors, so that tumor agnostic, and it serves, in principle, all patients, and particularly one category of cancers which are less well served by panels, even the large multimodal panels, which by definition and inherently can never be complete and don't give access to the tumor microenvironment. The category of patients who are the first one to benefit to CMTA are the rare cancers. Rare cancer are less than 6 cases in 100,000 in the EU, but collectively it's 25% of all cancers, all pediatrics. And they have the worst 5-year survival exactly for this reason, that the molecular diagnostic is not that well developed and that easy. It's one of the reasons. And also because they are rare, there are less trials, so there are several reasons to it. But what we address as well with the comprehensive diagnostics are metastatic and refractory cancers, which have developed additional abnormalities and complex abnormalities. So the rationale of pushing the type of diagnostics is to be able to serve patients lacking treatment option so far and being able to match more treatment and clinical trials. So in a nutshell, we have a single test which integrates genomic alteration with gene signature profile, which is powerful, broadly applicable for guiding therapy. And it's an accredited end-to-end diagnostic pipeline, which is also customizable. So we deliver a report which can be customizable to the needs of the hospital, of the clinics. It works from FFPE or frozen tissue. Frozen tissue is always better, but function as well with FFPE. And the turnaround time fits the patient journey. So how do we achieve that? So the pipeline is as I said, is a complete end-to-end accredited process. And from the tissue section, we extract the DNA and can then generate the NGS libraries and go to the sequencing. So the blue part is a wet lab part, and we do have developed proprietary pipelines in the bioinformatics pipeline to be able to translate this complex data into a concise report for the clinics, an interpreted report. So that is what that is now possible, which was more difficult some years ago, becomes possible for two reasons. You have the cost going completely down, being able to automatize the NGS libraries, and to be able to sequence in a short time at low cost. So now this is competitive to the multimodal panels in terms of cost. More in details, our modular workflow, we generally, when we do a full CMTA, we sequence about 20 gigabase to reach 250 times coverage of the WES of the tumors. The RNAseq is about 20 gigabase. So it can be adapted to the different needs. So what we use most often is a MGI G400 sequencer, and the reason for this is that it's very flexible from its format, and we can run two flow cells in parallel, each of about 360 gigabase for PF100 base pair, and we have 4 lanes, 4 independent lanes each. So it means that we can sequence lower throughput at low cost when necessary. The clinical samples, as you know, even from large hospitals, don't come in buckets of hundreds to super multiplex, so it's super practical to have a sequencer which deliver data at a medium output at very low cost. I will then detail a little bit our bioinformatics pipeline, which is the heart, I would say, the core of Alacris in some of the next slides. So in summary, the comprehensive analysis is now feasible and ultra competitive with other technologies because of the high flexibility of the instruments, and also library kits become more and more shorter and shorter, more and more practical. So with DNBSEQ technology that we have at Alacris, we can also sequence third-party libraries. What we observe is excellent quality, comparable to the competition, and a low duplication rate. So it's very practical for us and for our clients to sequence third-party libraries, because it is important to continue using the type of libraries that people have been using, and for some reasons it's always so many RNAseq type of libraries. It's good to be able to keep your own scheme. And what we have tested, for instance, we use very often the TrueSeq Ribosero library from Illumina, and what we compared here using the MGI G400 with our data on the NovaSeq 6000. So it's identical quality, lower duplication rate in the MGI, but by and large it's similar, it's identical. So what we did is to convert the TrueSeq Ribosero library. We use the same library, it's important to say, for the NovaSeq and for the MGI. The conversion is done with 6 cycles. The 6 cycles do not skew the data at all, and here you see that we have in read count normalized with count expression correlation, we have a Pearson correlation of 0.99. On this example, it varies between 0.95 to 0.99. So RNAseq, which is rather sensitive, variation is still completely comparable with the two technologies. What we see is that the complexity is higher with the DNBSEQ, which is due to the rolling circle technology, and basically the data are very, very comparable. For the coding gene, for the non-coding gene, we have an R of 0.72, but that's due to the fact that, as you probably know, non-coding genes are very low expressed. So if you are in the region of very low counts, you have a bit of a broader dispersion. But basically, yeah, we are very happy with the technology. So now I will walk you through for the rest of my talk to our pipeline, our CMTA pipeline, which combines exome-seq and RNAseq. So we use a number of open source and in-house scripts to be able to do that. So that's I could talk a long time about this, but we'll summarize in a few minutes. So we score the CNV, the chromosomal variants, and the loss of heterozygosity regions from the exome using the BLL frequency. We then integrate, when scoring the mutation, we integrate DNA and RNA calls. And that we find it important because we can, first of all, that's a way to filter potential spurious calls, and the mutations that are important are those which are expressed by and large, apart from the few examples of nonsense mediated decay for stop mutations. What we score as well are the gene fusions, the splice variants, transcript abundance, and we deconvolve the immune TME. So when we have all those outputs, which is the work of the callers and the integration between callers, then comes the filtering, because of course when you do all this is simplifies, you get hundreds and hundreds of calls. So we do have filtering steps which compile in the end the relevant true alteration, remove the noise, and we end up with a list of the alteration for that tumor. We also rank them by pathogenicity. And at the very last step, we do match our filter alteration with a database. So interpretation is always a long discussion. We can spend hours in discussing that. What we do is we match our filter data alteration with a database of drugs and clinical trials, and we use at the moment the QCI from Interpret One from QIAGEN, only at the very last step, and deliver a comprehensive report. So the report is schematized this way. I will not go through all of the features of the report. We list the targetable alteration on the first page, the TMB, the CNVs as well, and report the expression profiles, which are summarized here, which are the most important for that particular tumor, and the potentially the actionable variants for approved drugs and for clinical trials. The fusion, when there are some, are also reported with these schematics. So in a nutshell, what we have is a very comprehensive report. We can also do a HeLa typing when requested, which can be a long report. For some hospitals ask for a long report because they use it also for their research in addition. But for the tumor board and the clinical routine, we have a summary on 2 pages, which makes it easy to go through at the tumor board. So there is no need to choose which panel. We do extract what are the key alterations targetable for a given patient. The RNAseq deconvolution is also an interesting aspect, which is not requested by all of the hospitals. Some of our clients need this to evaluate some of the features of the tumor. And for instance, here is a deconvolution of a uveal melanoma, where, as it is well known for uveal melanoma, the story is not around PD-1, but more about LAG3, which is highly expressed. So that is explaining, well, showing rather than explaining how we can deconvolve the RNAseq data with an estimation of the respective fractions of the immune cells in terms of the M2 macrophages are also important to monitor for some of the tumor. So how to integrate and interpret DNA information? I think that I don't have time to really go into the details of our pipeline, but this combination allow us to deliver filtered results. So we don't rely on other systems. We have this in our pipeline, but we are preparing this to be able to deploy it to other premises, so to make it available to other institutes who would use MGI pipeline for sequencing and would need an analysis system. So we compile the tumor alteration and finally have a built-in annotation, and then the very final annotation to matching to the clinical trial can be the choice. One can use QCI, but one can use any other database of choice. I show you some of the selected features. I arrive at the end of my talk. And for instance, in this patient, we had detected focal amplification of EGFR, and we always deliver the expression value, which here show high expression by this green bar, together with the CNVs, because it happens that there is a CNV amplification, but the gene is not expressed. That has happened. So to get a higher level of precision for the diagnostics, we deliver the expression profile matching what we found at the DNA level. We do have a customer case that I would just mention in a minute. We do routine diagnostic for pediatric ILL in Germany, and we do this purely based on RNAseq. We don't do the exome, we do very deep RNAseq, and from the bulk RNA sequencing, we score the mutation, the gene fusion, on demanding gene signatures from the clinics. And that delivers a report which reduces cost and time compared to multiple tests which were previously done. Some of the selected features are TP53 and RAS mutation, typical for some of the ILL, and gene fusion. So for this, for the ILL, the report is only 2 pages. It's very short and go to the main alterations. So from the RNAseq, one can really, we don't necessarily need the exome if we are not looking for the CNVs. If it's deep enough, we can perfectly score the mutations. In conclusion, we have developed, like others develop as well, comprehensive profiling, which is at the forefront of a global change in the way the diagnostics and the treatments are being approached, which can support precision medicine-based drug repositioning implementation, because the data are there. So one can pick again in the data what is needed. It's a dynamic reporting adapting to novel actionable target, which the panel cannot do. Then what to generate the next panel. When one works with bulk, everything is in there. And so that we can meet today and tomorrow's customer needs. The CMTA is scalable reporting for different applications, tumor boards, clinical trials, and clinical research. In conclusion, we are navigating the space of diagnostics with comprehensive profiling, accelerate precision medicine solution, generate the deep data for accompanying the rapidly evolving field of personalized cancer medicine. Still, the interpretation of somatic alteration remains a challenge for all of us. The key is to identify the alteration and then to match them with the available database, but no one can go behind the available database. So of course, there are still a number of somatic alterations which are hard to interpret for anybody. However, having a more comprehensive test allows us to understand better the landscape of the tumor, better data, smarter diagnostics for the evolving health ecosystem, which deliver value-based accredited data, which allow data reuse, lowering the cost, a resource for enhanced data and clinical trials. And with this, I thank you for your attention and take questions if any.
Great. Yeah, thank you, Mari, for some insights into CMTA. So the floor is open to questions, and I've received some questions from the participants. So quite a lot of questions regarding CMTA. So Mari, I think one thing just to be clear on, is the CMTA solution currently available commercially right now? And if so, can you maybe comment as to who you have been working with?
With whom I've been working with?
Yes.
Yes. I don't know if all of our clients want to be quoted. Yes, I'm working, being in Berlin, work with the Charité Berlin. I also work with clinical units in Zurich. We also use the CMTA for clients who have sequenced themselves their data, because of course, since we have the wet lab and the bioinformatics, we can take already sequenced data, and we have clients in the UK and in the United States, mainly for neuronal cancer. So we have, I don't know if I would like to deploy all the list of clients without asking them their authorization. But for a number of units, yes.
And regarding the sample, so FFPE is an interesting one. How did you enable using FFPE samples? And how old can FFPE samples be?
Yes, that's a classical question. Well, if it's not so old, it's always better, but we have had good data, good for FFPE, with samples which were a couple of years old, right? If you want to go super retrospective, then it would depend of how the samples have been processed. But the truth for FFPE is you could also have bad data with a sample done the week before. What is really key is how the sample has been processed during the block. So the best would be to use cold formalin and adhere to very strict SOPs, which is done in some entities, but not always for a number of logistic reasons. So it works well, but the variation, of course, comes from how the samples have been preserved. Now, how we manage that, we do have a bit of a different extraction methods. And the quality will always be better with fresh frozen. But however, we do score fusion, we do score mutations. It works if the sequencing is deep enough. And as I said, if we have prepared RNA DNA with the appropriate protocol.
Great. And how did you validate the CMTA solution for accreditation? And is the list of accredited genes available?
Yes, it's on the website. Yes, sorry, I forgot to mention this.
And how did you validate the CMTA solution?
Yeah, that's a long story. Yes, I mean, when you go through the accreditation, you know, you have to go through a number of steps, and with your QMS, your SOPs, and we also run, of course, proficiency testing, and demonstrate that you find what you are supposed to find in terms of fusion mutations. expression profiles with other technologies to be able to benchmark. So yeah, that's a long way, but that we repeat regularly for the accreditation. And the bioinformatics, you know, same story. So you have two parts, the lab and the bioinformatics. And the bioinformatics is a mix of proficiency testings and benchmarking with data coming from other labs.
And regarding the turnaround time from sample in lab to report, can you just comment on that? And how does adopting MGI influence the speed?
So I will start by the second question. I mean, adopting MGI influences the speed in the sense that we don't have to wait to fill up a very large flow cell to get to low cost. So that's the flexibility, having a possibility to sequence with you know, with medium throughput or even low throughput is influence the speed and the cost, because you can start sequencing before having collecting I don't know how many samples, right? What was the first question? Did I miss
the turnaround time from sample to report?
Yes, the TAT depends depends of the samples. We are about 2 weeks nowadays and try to cut it to 1 week. But the problem is, if someone sends us one sample, still sequencing one sample alone is, if it happens that it falls on a week when there are no other samples, then we have to wait to the next week, so the 2 weeks. But if we were to when we have the right let's say logistics, that is, yeah, a week, 10 days. Still the bioinformatics takes a bit of time, so between, let's say, 2 weeks is reasonable.
And this one is, I would say, maybe a mix between you and Priyanka. Obviously, the G99 was presented. Is it possible to run the CMTA solution on G99? Priyanka, maybe you can comment.
We need 60 GB for a full CMTA with the tumor and the blood reference and the RNAseq. It's about 60 GB. If we do a CMTA only based on RNA that we do for leukemias, then we use 26 to 28 GB. So Priyanka can calculate.
That's right. That's right. It depends on the capacity. So as you know, like for the G99, we have 2 flow cells and like 80 million reads that you would need. So yes, it just depends on the type of sample and the capacity that they require.
Cool. And then
output, of course.
Okay, and so just within the lab at Alacris, what are the other applications or solutions that are being run on MGI in the lab?
Out of diagnostics, we do metagenomics. We do recently we use the T7 for 10x single cell analysis, and that's great because we can get our 10x data quicker and cheaper on the T7. The run is overnight, and so that is really good for this. We also run VGBS, so whole genome bisulfite sequencing, VGS whole genome sequencing. Since we can convert the libraries, really there is no issue to convert the 10x. So that is a really very flexible system.
And just one last question regarding the RNAseq. Is it targeted or total RNAseq?
It's bulk. I don't use a targeted RNAseq because there's always something missing. And most of the clients we have who go through this more comprehensive solution, they like to have the bulk as well, because if they want us to reanalyze or do clustering of their patient cohorts, you can only do that properly with the bulk and also access the long non-coding RNAs. have all the splice variants, so we use bulk.
Great. And I think that's it for today. If anyone has any other questions, you may reach out to us on our genomics.com, genomicsunlocked.com website. And yeah, we'll get back to you soon. So just thank you again to Dr. Mari Law and to Priyanka for their presentations today. I think it was very insightful and we had a good number of questions. And yeah, we hope to see you at our next one. So keep well and goodbye for now.
Thank you. Goodbye.
Goodbye. Thank you very much.